Hi, thank you very much. Okay, well, thank you very much for the kind introduction. I'm very excited to be here today and to be able to tell you about the work we're doing at Isomorphic Labs to use AI to deepen our understanding of science and of biochemistry, but also ultimately to apply it to really impactful problems in drug discovery. Our mission at Isomorphic Labs is a really simple one that fits very neat in one single slide. We're here to solve all disease. I find when I first see this written down, it feels a little bit sort of, crazy, ambitious, and a little bit, outlandish perhaps. But I hope to be able to show you today that some of the methods we're developing are really making significant strides towards addressing this issue. Now, before I get into explaining about what we're doing, let me tell you a few things about our origins. Isomorphic was founded by Demis Hassabis, who's our CEO. He still leads Google DeepMind. And it's largely based on some of the work, the kernel of Isomorphic Labs was AlphaFold 2, a model to predict the structure of proteins. I myself used to work at Google DeepMind. I worked on AlphaFold 1, a bit before then. This seed that formed our company is also something that I nurtured back then in Google. We're a very interdisciplinary team. There's around 300 of us at the moment, and we're still a growing team. Here I show you our fantastic executive leadership team. And also, you can see we have a very eminent scientific advisory board that keeps us on track and makes sure that we're, really working on impactful things. Now, by training, I'm a physicist, and I'm quite used to the idea that the laws of the natural world, especially the laws of physics, are easily embedded in mathematics, can easily be described in a reductionist way, so to speak, and that maths really forms the natural language to describe the physical world. But in my opinion, I would say biology hasn't quite yielded to this same approach. It's difficult to reduce the complexity of biology to a set of reductionist laws. And it's really important, I think one of the things that's impressed me so much about AlphaFold 2 is how you can integrate experimental evidence with some ideas from physics, from chemistry, but you can integrate all this diverse information with data through AI. And one of our theses really is that AI is the natural language of biology, and therefore will form like a real basis for improving our understanding of this field. Now, this is important not only because it's an interesting academic question, but also because there's a really burning issue at play, which is the fact that the efficiency of pharma, of discovering drugs, decreases exponentially with time. It's getting harder and harder and more expensive to discover new drugs. On the other hand, we're well familiar with Moore's law, which says that the cost of compute is getting cheaper and cheaper every year. This fact that AI is such a powerful tool for describing biology, perhaps our hope is that this will be really a way to kind of complement the decrease in efficiency of pharma, so that by leveraging AI, by leveraging the fact that compute is getting cheaper, we'll be able to do more and more of biology in the computer. Now, before I tell you about our most recent results, I wanted to talk a bit more about the kernel that forms the basis for Isomorphic Labs, which is protein structure. The philosophy of drug discovery that we adhere to is of structure-based drug design. The fundamental entity that we work with are proteins, and proteins are kind of tiny little machines that are inside each of your cell. This is an example of one. This mint green blob is a protein whose job is to repair DNA. For instance, this particular DNA repair protein is the only DNA repair mechanism in certain types of cancer cells. And so, in a very simplistic way, if you're able to inhibit this protein, if you're able to stop it from doing its work, then you will severely damage the cancer cell that depends on it for its survival. So the goal of structure-based drug design is to discover small molecules which are able to precisely fit into a protein and manipulate its behavior, maybe stop it from doing its job in this case. In this particular case, the idea this is an actual drug that actually inhibits this DNA repair protein and thus has a therapeutic effect for cancer. Now, the problem with this approach, is that determining the structure of proteins and ligands together is a very, very experimentally difficult task that can require months and months of work. So the fundamental approach that we're taking to computer-based structure determination is basically to turn this problem into a machine learning problem, so to speak. The approach we take is conceptually not totally dissimilar to how language models work. We take the biochemical system we're interested in, and we describe it as a sort of sentence. So, for instance, a protein can be described by the letters that make up the amino acids. A DNA sequence can be described as a series of base pairs that make up the DNA, and molecules themselves can be tokenized in a similar way. Now, where we depart a bit from traditional language models is that instead of operating on tokens as a linear sequence, we operate on the relationship between tokens. We've invented an architecture called the performer, which essentially carries out attention on the interaction between tokens. This forms very rich representations of the fundamental interactions that are occurring in a biochemical system. And these rich embeddings can be passed on to a diffusion model, like model not that dissimilar to how some of the image generation methods like DALI or Imagen you might be familiar with, except that instead of producing an image, it produces a very accurate 3D structure of a biochemical system. So I've been able to tell you now about the kernel that has formed the basis for our company in some way. But today I'm really excited to be able to go a little bit further and to share with you some of the results that have just come out today in a technical report that's on our website, and which describe our next steps. Our next steps in developing a more comprehensive drug design engine, which is capable of performing several tasks. I'll be able to share some of the particular aspects of this drug design engine and tell you some of our interesting results. Now, I'm going to start off with structure. Structure is still a very challenging problem, especially when we try and work on ligand protein structures which are very different from the training set. The way that this is usually measured is through a public benchmark known as runs and poses, where examples are stratified by how similar they are to the training set. And on the most dissimilar, on the most novel ligand-protein complexes, most sort of frontier AI methods, including our own AlphaFold 3 from 2024, really struggle and have an average sort of success rate of around 20–25%, whereas ISO's latest drug discovery engine is almost twice as accurate. Now, we don't only work with small molecules, however, and we also are interested in other modalities of therapeutics, an example of which are antibodies. Antibodies are yet another important protein in your body, and their antibodies' job is really to recognize specifically certain other proteins. As well as being produced by your body, they can also be used as therapeutics. Now, antibody-antigen structure prediction is the problem, the equivalent problem of finding the relative shape of a small molecule and a protein. It's about finding the relative shape of two proteins in some sense, and it's a very, very challenging problem in drug design. Again, I'm sharing here, the most challenging benchmark we run on this problem, which is this high accuracy success rate for antibody-antigen pairs. And again, we can see that our latest models are not only more accurate than AlphaFold 3, but vastly more accurate than other open source models. Another important problem in drug discovery is finding pockets on a protein. The pockets are the places where a protein can be liganded. They're sort of the keyholes in which we're trying to find specific binders. And unfortunately, in the protein database, a very small fraction of the proteome is actually present with liganded ligands. So if one really wants to fully identify all of the pockets on a protein, it has to be done experimentally, which takes months again of experimental time and is very costly. On the other side, our drug design engine is able to, recapitulate to a high degree of accuracy pockets which would be found experimentally, but in a matter of minutes. And the fact that you're able to do this on a computer opens the possibility to finding pockets at scale, which opens the possibility for being able to identify, all the pockets in the entire proteome, which is a very valuable resource for our drug discovery scientists. Now, so far I've talked about structure, but structure isn't the only problem you need to solve for drug discovery. For instance, it's very important to know for particular ligands, not only where they fit into the protein, but also how hard they stick. This problem is called binding affinity determination. Again, ISO's drug discovery engine, we've comparing it here to a bunch of other machine learning models, but also to physics methods based on molecular dynamics. This is kind of my, before joining DeepMind, this was sort of my area of expertise, was atomistic modeling. And to me, it's really incredible that you're able to perform as well or even better than physics-based methods at an absolutely tiny fraction of the computational cost. So I've described so far a suite of methods which are able to characterize therapeutics and their interactions with proteins. But this isn't quite enough for really designing therapeutics. We also develop generative models that are able to work with different modalities. They're able to take desired properties, desired binding affinities, desired modes of binding, and design small molecules or peptides or even glues or antibodies which have the desired characteristics. And again, we don't just do this, what excites me about a place like about ICE is that we're not just doing this, on a sort of benchmark in the computer, so to speak. We're also doing this, for real in drug discovery projects. In this figure, for instance, I show our hit rate for de novo small molecule generation, showing that on a suite of our internal projects, we're able to achieve high fractions of active developable hits, which surpass the industry average by a considerable margin. Likewise, we've run discovery campaigns on antibodies, and we're able to recapitulate on really challenging epitopes, on really challenging antigens, a high fraction of hits which are specific to the desired target. So we're able to develop antibodies that can be developed into real therapeutics using our drug design engine. Now, I've given you some idea, I think, about the principles that drive the design of the models we're working on. We really want to work on methods which are generalizable, that really try and do novel things, that are not simply recapitulating what is known in the training set, but really are usable in a discovery context to extend knowledge outside of what they've been directly trained on. Our methods, are in the computer. They're incredibly scalable compared to experimental methods, and therefore, together these characteristics mean that we're able to work on multiple modalities in drug discovery. We're able to work with different types of therapeutics so that no patient is left behind, and so that we make significant steps towards solving disease. And with that, I'd really like to thank you all for your attention, and thanks the organizers for their kind invitation.